because of this drug combination situation, recently there are some papers suggesting that, like currently with cancer therapy, usually what you do is if you have a new drug, the phase 1 clinical trial just do different doses to see at what level the patient can still tolerate without too much toxicity. The phase 2 clinical trial is to show to compare this with placebo to see that whether the drug alone can work better by itself compared to just placebo. And phase 3, usually you do the clinical trial comparing this drug alone with the current state of the art, say comparing with chemotherapy or with another targeted therapy, and to see whether it does better. And so, but then very often you treat the patient with a single agent therapy, and when they become resistant later, initially the patient might respond better, and when they become resistant, then you try another therapy to overcome that resistance. And this particular paper is suggesting that sequential therapy seems to be a perfect recipe for certain treatment failure, because if you just treat with one therapy, over time it will become resistant, and then you treat with a second therapy, it will also likely to develop resistance, and then you treat with a third. Instead, if the patient, the suggestion is if the patient can actually tolerate the toxicity from the combination, you should treat the patient with a combination from the start. You do not allow it to have the chance to escape and become resistant, right? Which means, successful combination of therapy must force the cancers to make at least two mutations or two evasion mechanisms at the same time in order to become resistant. It's much, much harder. Therefore, if the patient can tolerate the combination therapy, potentially treating them together is a much more effective treatment, which questions whether the current FDA clinical trial is ethical, because that the current trials is asking this new drug has to work by itself. Maybe it doesn't, but it doesn't mean the drug is bad, right? If it can use in combination with other drugs, it's still nonetheless a very, very good therapy. And potentially you want to treat the patient with two drugs from the beginning rather than waiting for the patient to become resistant. Yes, Jed?
How are combination clinical trials currently done now?
It's not done as much in targeted therapy. It's done a lot more in immunotherapy, which is great. I think immunotherapy also, interestingly, previously, in order for any drug to work, you have to really figure out the mechanism, test it in mouse, in monkey, before you try it in human. But interestingly, I think the immune system between human and mouse are already very, very different. So, if you ask the current, the PD-1 companies like Merck and Bristol Myers Squibb, they are really starting to try PD-1 antibody with another targeted therapy, with chemotherapy, with some other drugs now. Even though it may or may not work as well in the mouse, because the mouse immune system is just very different. In other targeted realm, it's been a little harder. So chemotherapy was first developed by Sidney Farber, who started Dana-Farber Cancer Institute. Initially, he just tried one drug, and the patient, in very quick time respond. Magically, all the tumor symptoms went away. These are leukemic patients, pediatric leukemia patients. And then after a few months, or even a couple months, the patient would relapse and come back and eventually die. And the first place to try combination therapy was in National Cancer Institute. They decided to try 4 drugs at the same time. And at the time, people thought they were crazy, because if one drug is toxic, 4 drugs together will really be so toxic. And they just said, okay, let's try it. Maybe the idea was maybe every drug only kill 99% of the cell, and another drug will kill 99% of the cell. But hopefully the 1% from one drug and the 1% from the other drug is a different 1%. And so maybe you add the 2 drugs together, they will work better. And so they tried the 4 drugs together. And initially, indeed, the patients were extremely sick, and there was nothing the doctors can do. They just fluff their pillows and wait, right? And then magically those patients indeed got cured. And now chemotherapy, at least for leukemia, is always a cocktail. And if FDA was around at that time, those drugs would never have happened. The chemotherapy would not have made it. And so, now it's like you have to make sure each one work, and then you try the two, and then you try the third by itself, try the third with the first, and the third with the second, and then try the three together. It's crazy, like how many combinations you have to do in order to make a 4-combination cocktail. It's just not feasible. And so I think FDA is re-evaluating, is rethinking based on the current understanding of the cancer mechanism. I think we're going to see some policy changes now. Okay, so in general, I think in the long run, you will see more and more personalized therapy, combination therapy as well. Targeted therapy or chemotherapy, I think it's still one major, like currently still most patients get treated with either chemotherapy or targeted therapy. They work by directly kill the cancer cells. Some of them, of course, also kill your normal cells as well. Immunotherapy is not really directly working on the cancer cell. They work on kind of trying to boost your immune system and let your T cells and B cells or natural killer cells or whatever to kill the cancer cells indirectly. Epigenetic therapy is just starting. People are just starting to try it. It seemed to work a little bit. Some of these trials recently are failing. They might work well in animal models. They might also well in cancer cell lines, but so far in the clinical trials, the success is still very limited. As we imagine, epigenetic therapy is kind of just tweaking the system balance or the plasticity a little bit. It's hard to really, when you tweak something, it's hard to make sure that it happens to tweak in the right way. But potentially, as we mentioned, every time we do a CRISPR screen on a drug treatment, you see these epigenetic genes showing up, suggesting that maybe as a single agent drug, these are not good, but as combination therapy, they can become much better. It just reduces the plasticity or allow the immune system to turn on a little bit better. And so in order to really do these combinations well, we think, tumor profile will still be very, very important. You can imagine doing like DNA or RNA. You can even do single cell RNA-seq, right? We can also do CRISPR screens. We mentioned that in the last lecture, how to really, look at, drug resistance mechanism or look at drug combinations. And of course, all of this generates a lot of data, which takes computational modeling or analysis. And but that's how you can really effectively combine the different drugs. What I can imagine is, in the future, once you have the patient coming in, based on the whatever molecular profile you did on the tumor or on the tumor microenvironment, you decide, give them either a personalized cancer vaccine, the drug, like the different new antigen vaccines, or you give the patient, this epigenetic drug plus that targeted drug plus this particular epigenetic immunotherapy combination. When then another patient comes based on the the tumor profile, you will give them a different combination. I think that's definitely going to happen. Maybe not quite 2020, but I think within the lifetime of most people in the audience, we will definitely see more personalized combination therapy. But in order to do those well, they will need to do more of these high throughput technologies and also to know how to do computational biology well. Okay, so I think that's the third module. In the next lecture, I will start to give you some examples of what, we'll do the third module review and also give you some examples of what the final exam might look like. And next Tuesday, unfortunately, I will be traveling again. There is a video I hope all of you guys watch. It's one of the biggest scientific fraud in US, especially cancer. And potentially a lot of patients get harmed. It's a pretty serious issue. At the same time, that lecture is extremely entertaining. You should see how a computational biologist went through the detective work to figure out that the other group was cheating on their publications, not in order of one, but in terms of like 10, 20 different publications. It's another computational biologist, like manipulating the data and how, it was gradually discovered, which I think we should all make sure. So right now with a lot of journals, making sure that the results are reproducible, the code is there, the data is made available is now a very big deal. And I hope all of the students in this class learn to conduct reproducible research. Make sure you document your code, you keep version control, you upload your code for like GitHub for sharing, right? We will continue in the next lecture for the final review. Okay, thanks.